Hi, I'm Nicole, and this is a Hawaiian bobtail squid. The light organ in its head glows in the dark, or in more sciencey terms, bioluminesces at night. Where does the light come from, you may ask? Well, it's actually produced by bacteria, the species Vibrio fischeri. They are involved in this process called quorum sensing. We can think of it as like this big group chat where bacteria send and receive chemical code words to and from one another. We generally call these code words autoinducers since they're automatically being typed and sent out by the bacteria. But in reality, it's more complex than that, and to see what's actually happening, we have to look at a certain set of genes in their DNA called the LUX operon. The operon has the genes that can make bioluminescence happen through a chemical reaction. Here's the catch. The operon isn't always on or active. It can be, but only if there are enough bacteria around to produce the needed amount of autoinducers. Using our analogy from earlier, we need enough bacteria in the group chat constantly sending each other code words in order to make plans as a big group and do activities together, like glowing in the dark. As we can see here, LuxI encodes for the enzyme that makes the autoinducer. In this case, it's one of a class of chemical compounds we abbreviate as AHLs. LuxR, on the other hand, encodes for an activator protein, which activates the operon, but only if an autoinducer binds to it to form a partnership we call a complex, and that said complex binds to the Lux box, which kinda acts like the switch you need to flick on. Since there aren't enough autoinducers present, no binding virtually happens, and that both autoinducers and activator proteins are still just being produced. On the flip side, when there are a ton of autoinducers around, one will bind to an activator, and this autoinducer activator complex will bind to the Lux box and turn on the operon. Lux, C, D, E, and G produce the proteins that create or convert the chemical substrates needed for the chemical reaction. Lux A and B produce the alpha and beta subunits, respectively, of the enzyme luciferase. Luciferase catalyzes the reaction involving the substrates, which ultimately creates a light byproduct. This light then illuminates the bacteria in the light organ, which then allows our Hawaiian bobtail squid friend to glow in the dark.